Hello everyone, I'm Rafaela and today I'm here with the iGEM team of Aachen. Today's video is all about energy. We know that every living organism, from bacteria to complex animals and plants, needs energy to keep itself alive and functional. But what exactly is biological energy and how do we transport it in our body?
Inside the cell, energy is constantly being transformed by thousands of chemical reactions, which are often linked together in chains or pathways. The entirety of these reactions is named metabolism. Metabolism is one of the most crucial processes that define life, just like having DNA or cellular form. Metabolic reactions can be either happening spontaneously under the release of energy, or they need to receive energy in order to take place. Energy-releasing reactions, for example the breaking down of sugars or the nutrients, are also called catabolic reactions. Energy-receiving reactions, like the synthesis of cell proteins and many other enzymatic syntheses, are called anabolic reactions.
In a cell, the catabolic reactions are the only source of energy for anabolic reactions. So between them, there has to occur some kind of energy transfer. This energy transfer happens by the synthesis and hydrolysis of the molecule ATP. ATP is sometimes also called the cell's energy currency because of its ability to easily take up and release energy.
But why can't energy just directly flow from catabolic to anabolic reactions? That's because every biochemical reaction is catalyzed by enzymes, which make sure that the transfer chemical energy doesn't get lost on the way between the reactions, for example, in form of heat. An enzyme can only catalyze very certain reactions, since the molecules, also called substrates, have to fit perfectly into the specific binding site of the enzyme. So there will be an incredible amount of different enzymes necessary to connect every catabolic to every anabolic reaction. However, with ATP as an universal energy carrier, there just has to be one extra binding site on each enzyme.
ATP stands for adenosine triphosphate, and it's also one of the 4 nucleotides your RNA is made up of. It's by far the most commonly used energy carrier, and it's universally found in every living being. But why aren't there any other molecules like ATP? It's believed that ATP became an energy currency very early in life history. And once the currency gets established, it would be just too unlikely to be replaced by another one.
ATP consists of the base adenine, the sugar ribose, and 3 phosphate groups. The bonds between the phosphates are called phosphate anhydride bonds. Especially the outer bond is very rich in energy, so in order to get into a more favorable energetic state, the outer phosphate group really wants to split off from the rest of the molecule. This reaction results in the molecules adenosine diphosphate, short ADP, and phosphate. It also consumes 1 molecule of water, that's why it's often referred to as hydrolysis.
The energy, which gets released through the hydrolysis, can be used to power any anabolic reaction. It is important to say that without the help of ATP, anabolic reactions wouldn't occur at all. That's because the reaction doesn't want to happen, just as much as the phosphate group from ATP wants to split off. Now we come to the synthesis of ATP, which is just the exact opposite of hydrolysis. ATP is formed again out of ADP and phosphate, but as we remember, ADP and phosphate doesn't want to live together. Therefore, a certain amount of energy is needed to enforce them. This energy is provided by catabolic reactions.
In the field of synthetic biology, especially anabolic reactions are quite important. That's because they are responsible for the synthesis of basically every biochemical compound. So the constant supply of ATP is one of the main reasons why many products like insulin, antibiotics, or certain enzymes are produced using living microorganisms. However, some products are harmful to microorganisms. For example, phenylacetylcarbinol, which is a precursor of many pharmaceutics. Therefore, it has to be synthesized in a so-called cell-free system. In cell-free systems, enzymes and ATP have to be added. While the enzymes can perform the same reaction over and over again, ATP becomes useless after reacting to ADP. Unfortunately, ATP is quite expensive. So cell-free systems are only suitable for very small quantities. Also, the industrial production of ATP consumes a lot of resources and causes avoidable pollution.
And that's the point where our iGEM project MARS comes into play. MARS is short for Magnetic ATP Recycling System. Our goal is to create a bioreactor in which ADP is constantly being recycled to ATP. The recycling is done by our chassis, which are tiny protocells that contain two different enzymes. The first one is bacteriorhodopsin, which create a proton gradient by only using light energy. The other one is the ATP synthase, which can then use that proton gradient to synthesize ATP out of the ATP and phosphate. These chassis are also attached to magnetic particles, so they can be magnetically separated when the product gets harvested. After that, the chassis can even be reused for another cycle. This makes ATP production with MARS much more sustainable and cheaper than any previous methods. Thank you for watching. Don't forget to subscribe and stay tuned for more energizing videos.